I've heard people saying that components of SR2 they are not at one place, they are scattered. What does this mean?
In the genome?
I mean, it's not at, SR2's, what would I say?
You're talking about the SR2 complex?
Yeah.
Yeah. Okay, so it's a little bit off track from what we're talking about now, but I'm happy to discuss that. You'll read in the literature and you'll hear people talk about the SR2 complex. We know that in some varieties they carry if you take a variety that carries only SR2, it's moderately susceptible at worst, right? You know another variety that carries SR2 and it's moderately resistant or resistant. So obviously it's got something in addition to SR2, but we don't know what. And what they've found is often those additional genes are of minor effect, and there are a number of them, and they've been difficult to characterise, and so they've just referred to them as the SR2 complex. It doesn't imply that they sit in the same region in the genome. They could be right across the genome. It just means that there are these additional factors that are adding to the effect of SR2.
So it means there are minor genes apart from SR2?
Most likely. But interestingly, one of those has turned out to be SR12, and I predicted that many years ago. I said when they start looking at the SR2, I don't think I was the first person to say it, but I think people had forgotten about that, and I reminded them. I said SR12 is a major gene, really hard to phenotype in seedlings. I'll bet that comes up when they start doing QTL analysis, that region, and it did. So SR12 is one of those genes. It's a major gene, but it's a major gene that's really hard to phenotype because it doesn't give a strong level of resistance in seedlings. I know that sounds confusing, but... Sorry, Gordon.
Well, the paper by Liu, the one that compiles all of the UG9 refers to that locus, and there are apparently a number of QTLs that are clustered around that locus.
SR12 or the...
SR2.
Oh, SR2, okay. So yeah, they may or may not be associated with SR2.
Is there any example of codominance for rust resistance?
Codominance?
Is there any example that the genes affects codominant in rust resistance?
So you're referring to heterozygosity versus homozygosity?
There are characters that if two genes are together,
yep,
both of them might be interfering each other, resulting in no resistance. Is there any example?
Yeah, suppression, in other words. Suppression of resistance genes. Yes, we do have examples of that. The examples that I can think of suppression, the best example I can think of suppression is actually mildew resistance in wheat. And that's been published quite recently. But what they've shown is that there's a powdery mildew resistance gene in wheat, and it's actually the one on the 1B1R translocation with SR31, right? And if that's present with another powdery mildew resistance gene, you get suppression. So instead you have two resistance genes present, but instead of getting resistance, you get susceptibility. One suppresses the effect of the other. And they've actually been able to show why that is. And the reason is that the one in wheat and the one in rye are actually orthologs. They're evolutionarily related. They came from a common ancestor before wheat and rye evolved. So they're orthologs of the same gene in different plant species. We've brought them together, and there is some post-transcriptional event that's causing suppression of the gene products. Okay. Let's say that cultivar A has adult plant resistance to stem rust. How do we determine its genetic basis, right? We want to know whether it's dominant, whether it's recessive. We want to know how many genes there are. I mean, that's pretty important. The adult plant resistance, we might test cultivar A, or Zach might test it. He's knocked out SR31 and it's still highly resistant to stem rust. It's got a very high level of adult plant resistance. We want to know how many genes, and we want to know what they are. How do we do that?